Hello, I'm Jason Hatton. I'm responsible for the coordination of biology and exobiology in the SciSpace team of the Human Spaceflight and Robotic Exploration Program. So I'd like to give you a little bit of an overview of the research that we do using ISS. So the International Space Station, as its name suggests, is international, and basically it allows you to do very long duration experiments in the spaceflight environment. So looking at the effect of microgravity, low gravity, also exposure to the direct space environment. You can do research in all fields. You can study, of course, the astronauts, how they react to the space environment. So studying the changes in their physiology. You can study biology. So the cells, which would be cells from, say, human body, microbes, plants. You can study also physical sciences, materials science, fluid physics, and also, of course, you can look outside from the space station. You can look at the Earth, so you can do Earth observation. You can do also observation of the space environment. Nice thing is, because it's an international platform, all of the facilities, all of the instrumentation is shared. So that allows you then to do a lot of research and very internationally. So there's been about 3,000 experiments done over the lifetime of ISS. And those gave about 2,000 scientific publications. One of the areas, of course, of interest is astrobiology. Do microorganisms or microscopic animals in dormant state, can they survive exposure to the space environment? This is very important to see whether life could be transported from one planet to another on an asteroid, or whether also if you have, for instance, microbes on a spacecraft going from the Earth to Mars, how do you prevent contamination? The other aspect is also, how did life originate? So there's a lot of studies on the chemistry which led to life, so which may have been going on the primitive Earth or on the surface of a comet. So we developed an instrument called EXPOSE, and this was put on the outside of ISS and allowed exposure to direct space environment for typically 18 months, 2 years. And what was found was, in fact, that many of the microbes and even simple animals such as tardigrades, these are water bears, could actually survive direct exposure to the space environment for 18 months or so. And so that was a very interesting finding. The other area, of course, is plant biology. And there was actually a whole series of experiments done over a 10-year period by a team of European and US scientists. And they looked at all sorts of aspects of plant biology. They looked at how plants responded to gravity, because what you can do on board of the ISS is you can effectively look at what happens in the absence of gravity. You can apply, using a centrifuge, artificial gravity, and then see what is the perception threshold for plants to gravity. And that was actually found to be, hundreds of normal Earth's gravity. So plants are very sensitive. One of the experiments which was particularly nice was an experiment which studied a process called circumnutation. So when a plant grows, if you look very carefully, it doesn't just go straight up, it does a sort of circular motion. And with this circular motion, this was actually observed by Charles Darwin. Although he observed this, he couldn't figure out what the mechanism was. You always have gravity on the Earth. But going to the space station, it was possible to do an experiment where we could take away gravity, we could apply gravity, we could turn the light on and off, and answer this question. In fact, what it was found was it is internally driven, but it's very strongly affected in terms of the period and the intensity by gravity and by light. So that was a very nice result, answering a question that Charles Darwin had posed over 100 years earlier. A lot of the research which is done on board of ISS is basic research, but it has relevance to a number of applications. Material science, for instance, you can do much more precise measurements. Also research which is relevant to health. Right while I'm standing here, my body is working against gravity. And of course, in spaceflight, in microgravity conditions, you don't have that. So what happens is, unless you do exercise or some kind of countermeasures, so that could also be with diet, you will lose a lot of your bone and muscle mass. And this actually parallels some of the changes which occur when people are very inactive, so sitting at the desk, or in the elderly also have trouble with mobility. So you can look at, say, the transfer between the techniques you use to as what we call countermeasures against bone and muscle loss for astronauts, and what you would do for people on the Earth who also have problems with bone and muscle loss. You can also do research with things such as nematode worms. So there's small microscopic worms. They also have muscles, and they lose muscle mass in spaceflight. So, and it turns out that the genes which control the muscle proteins and the muscle growth are actually very similar to the genes which humans have. So you can actually study, you can manipulate that process, and that gives you some clues into the mechanism. So there's a whole series of experiments which have been done on that. The final thing is, of course, if you're going to Mars, you either take a lot of tin food with you, or you grow your own food during the mission to Mars, and also perhaps to regenerate your atmosphere, converting CO2 back into oxygen. So there's a project we have called Melissa, which is a bioregenerative life support system. So it has plants in there, it has algae in a bioreactor, it also has a waste treatment system. So all of the technology which goes into that also has applications on the Earth. If you want to do agriculture in a very intensive environment, so in a greenhouse, you want to do that very reliably. So that's also an area where there's transfers. So there's many different transfers of knowledge which are possible.